friends, welcome to my YouTube channel Sam Biology Class. Today we will discuss about restriction mapping. But before that, will you please consider subscribing my YouTube channel? Your subscription and viewership are often a lot to me. Restriction mapping is a physical mapping technique which is used to find out the specific locations where restriction enzymes cut a DNA strand. We know that restriction enzymes, also known as restriction endonucleases, are enzymes that cut the DNA at or near specific sites known as recognition sites or restriction sites. This technique helps us to draw a physical map known as restriction map that shows the relative order of different restriction sites and the distance between them. Steps of restriction mapping. First step is the determination of the shape of the sample DNA. In this step, we have to find out whether the DNA sample is linear or circular. For linear DNA, a single cut will produce two DNA fragments. So for linear DNA sample, we have to apply the rule, that is total number of fragments. That is total number of fragments is equal to n plus 1, where n is the number of cuts. So if there is a single cut, then we will get 1 plus 1 equal to 2 DNA fragments. 2 DNA fragments from a linear DNA sample. On the other hand, if the DNA is circular, then a single cut will produce one linear DNA. So for circular DNA molecules, n is equal to number of fragments. N is equal to number of fragments. Second step is restriction digestion. Second step is restriction digestion of separate DNA samples by one restriction enzyme alone, and then by combining two different enzymes. The third step is separation of DNA fragments. Separation of DNA fragments obtained from restriction digestion by different enzymes by agarose gel electrophoresis, and their size determination. Next step is analysis of DNA fragments. Analysis of DNA fragments based on their length. And the last step is arrangement of DNA fragments in an overlapping manner to determine the exact order of different restriction sites and the distance between them. Here is one example. We have been given a DNA sample whose length is 6.5 kb. The length of the DNA sample is 6.5 kb, And in this example, we have taken two different restriction enzymes, EcoR1 and HIN2. When the DNA sample is cut by EcoR1, two fragments are produced, out of which one is 3 kb and the other is 3.5 kb long. This indicates that the sample DNA is linear. In another experiment, a separate sample of the DNA is cut by another restriction enzyme, that is HIN2, which also produces two DNA fragments. One is 2 kb and the second one is 4.5 kb. In third step, another sample is digested by combining EcoR1 and HIN2. This is known as double digestion, which produced three fragments. One is 2 kb, second one is 1 kb and third one is 3.5 kb long. So now we got the length of DNA fragments that are obtained from different enzyme treatment after separating them by agarose gel electrophoresis. Now name the fragments obtained from double digestion, for example, as X, Y and Z. Following this, draw a table which shows the fragments obtained from single digestion is equal to the length of which fragment or fragments obtained from double digestion. First, from EcoR1 digestion, one fragment is 3 kb, which is equal to the length of fragment X plus Y. The second fragment is 3.5 kb, which is equal to the length of fragment Z. Next, from HIN2 digestion. From HIN2 digestion, first fragment is 2 kb, which is equal to the length of fragment X. And the second fragment, that is 4.5 kb, is equal to the length of fragment Y plus Z. So 4.5 kb is equal to Y plus Z. Following this, arrange these sequences in an overlapping manner so that the relative order of restriction sites can be determined on the DNA strands. How to arrange these sequences? Start with X. Then the overlapping sequence will be X plus Y. Then below write down X plus Y. Here there is overlapping. Following this, the next overlapping sequence will be Y plus Z. Y plus Z. So here there is overlapping. And the next overlapping sequence will be Z. Based on this overlapping, we can place these three fragments in a correct order. So if this is the DNA strand, first fragment will be X, second fragment will be Y, and third fragment will be Z. So the fragment Y is placed between X and Z. The length of fragment X is equal to 2 kb and that of Y is equal to only 1 kb and that of Z equal to 3.5 kb. 3.5 kb. So now the point is, where are the restriction sites for these two enzymes? This is the restriction sites for EcoR1. So when the DNA strand is cleared by echo R1, two fragments are obtained. One fragment is 3 kb and the other one is 3.5 kb. Now let us know the second restriction site where hint to cleave the DNA strand. From hint to digestion, one fragment is 2 kb and the other fragment is 4.5 kb. So this is the restriction site for hint 2. When the DNA is cut by hint 2 and from double digestion, three fragments are obtained. One is 2 kb, middle fragment is 1 kb and last one is 3.5 kb. And this is the restriction map for the DNA strand. For other examples also, we can apply the same procedure. Restriction mapping is a technique used for drawing restriction map of the sample DNA, sometimes for DNA sequencing and comparing the DNA samples obtained from different sources. So friends, this is all about restriction mapping. To get more such updates, subscribe my YouTube channel. Goodbye and good night.